Guys, you can start. I think there are 100 people.
Great. Hello, everyone. I'm David Hyton from Brisbane, University of Queensland. And thank you, Mari, for asking me to chair the session. I'll be chairing the introductions, and then Pepe will be managing the questions at the end for the last 20 minutes. So as Mari mentioned, we will start messaging you and turning you off after 5 to 6 minutes. So it's my pleasure first to introduce Turgut Durderin from the Institute of Photonic Sciences, Spain, and he's going to talk about hybrid diffuse optical technologies for neuromonitoring in the clinics. Thanks, Turgut. Do you want to share your presentation? Great.
Hello, thank you. It is really great to participate in this. I mean, it's been a great 2 days that has been going on. And when we were planning this, I wasn't sure what exactly to prepare. And even though we saw a variety of talks from last 2 days, I have decided to just focus completely on the clinical part and explain about a couple of relatively large scale clinical trials that we are involved in. So as you know, these type of technologies, NIRS and DCS, can be relevant to many, many applications, but not all applications are mature enough to be studying large number of subjects. So I don't know whether my screen is properly showing. I cannot see half of it. Let me see if we can minimize this.
Okay, it looks good.
It looks it's good. Fine, Turgut. It's fine.
Perfect. One of the clinical trials we are involved in is called Stand Up. It has been spearheaded by Raquel Delgado Mederos at Hospital de la Santa Creu in São Paulo. She's been particularly interested in taking our previous work on obstructive sleep apnea, internal carotid artery stenosis, and acute ischemic stroke to the next level to see if the type of measurements we do there can be used to whether we can study early mobilization therapy effects and whether we could use some of these measurements to guide it. The point here is that early mobilization therapy after ischemic stroke has failed in some big clinical trials, but it is also a very promising technology on the other hand. And what they are seeing is that personalization and picking up a more heterogeneous sample of subjects that fit this goal will work. So she's looking into basically by doing this head of bed challenges that we use in stroke and other high risk patients as a way to evaluate the ability of a subject's brain to tolerate early mobilization. It's still ongoing. The goal is to measure more than 200 subjects, and we have measured about 70. So no results yet. In another study called Front Stage, a geriatrician called Marco Inzitari from Park Sanitari Pere Virgili, with whom we have carried out a preliminary work with about 70 subjects before, is interested in characterizing the prefrontal cortex function in cognitive impairment and slow gait. And in this case, we have 3 cohorts that we will study. One cohort will receive physical exercise as a therapy. We'll look before and after this therapy and correlate optical measurements, FTCS and FNIRS, to the cognitive outcomes. Other group will also receive transcranial electrical stimulation by the project partner Neuroelectrics. And the third group will be basically healthy aging and risk factor groups. So this study has just started to recruit sort of the pilots group, and it's going to progress very rapidly over the last coming year and a half. And we'll be looking at both cognitive and gait-related tasks to study. We have also recently announced a joint laboratory between ICFO and one of the largest children's hospitals in the region, San Juan de Deu. And with them, we have 3 projects ongoing. 2 of them are related to the functional NIRS and functional DCS, mainly time-resolved spectroscopy and DCS. In one of them, we'll be looking at babies born with congenital heart diseases. And in the other one, we will be looking at children having seizures. I should also point out something very exciting has happened, and we will start a new project called Tiny Brains, possibly reflecting the size of my brain, in early December, where we'll have industry as well as us participating to make sort of a monster DCT, DOT high density device to do the similar studies on the babies. This being a joint lab will sort of seed the creation of multiple projects, and we have a lot of excitement going on. And fresh out of the oven, we have started to work on COVID-19 subjects. So Around April, became very clear that COVID-19 also has a vascular effect. In fact, it causes these ACE2 receptors and endothelium are affected by the virus. This leads to the formation of blood clots and other problems. And this, combined with our desire to find some relevance when we went into a lockdown, led to a discussion with Jaume Mescuda. You see his picture on the bottom right, from Partoli Hospital Universitari. He used to use NIRS on the muscles of sepsis and other patients in the intensive care unit. And he was, let's say, brave enough to start talking to us while he was seeing patients 24 hours a day, 7 days a week at the height of pandemic. And this turned into big collaboration. Some of our collaborators are actually from this community, involving Spain, Brazil, USA, and Mexico, with 9 hospitals and 2 academic centers. Other than ICFO, we have Centro de Recerca Matemática. They'll be doing the statistical modeling. And in this, our goal is to recruit 800 patients and 400 controls for the first part of the study to study endovascular dysfunction in COVID-19 and non-COVID-19 patients. And in a sub-study of 250 patients, we'll look at the short-term effects of prone positioning in the same group. This collaboration is part of the Open Science Initiative of EU as well. So we will be releasing data often, and we are accepting new members. But we didn't stop there. European Commission had a call for emergency funds to meet the pandemic. And we have recently been awarded with a project called WASCOVID, which sort of complements the HemoCOVID in the sense that ICFO and Politecnico Milano will develop a battery-operated wireless DCS-TRS machine, work with BioPixels to develop phantoms and introduce standards, which will be very relevant to take this to large scale. And we'll work with a Dutch company, SplendoFit, for artificial intelligence and machine learning in order to extract the most relevant data from this muscle data we'll get. And Hemophotonics, which is a spin-off of ICFO, will participate in the project for integration and commercialization, as well as Aspholion, which is a regulatory affairs company, which will allow us to go for CNFDA certification and will carry out clinical trials. And finally, since we have been having a lot of different projects all over the place.
Turgut, you are at 7 minutes.
I'm done.
Thank you very much, Turgut. Now, I think next, are we having Mario Forcioni rather than David Davies? I don't think I think David Davies is occupied in theatre at the moment. Are you there, Mario?
Yes, I am. Yes.
Yeah.
Can you hear me?
Yeah, thanks.
Okay.
So Mario's from the University Hospitals Birmingham. Thank you very much for coming. He's going to talk about applications of optics in brain trauma care. And we'll give you a warning if you reach your time. Thanks.
Thank you. Apologies on behalf of Dave Davis. As he said, he's going to, he's currently in theater, so I'm going to talk on his behalf. I'm going to give a quick tour on what is our research group and where we are working. So we are working in the Queen Elizabeth Hospital in Birmingham, in the United Kingdom. And we have one of the biggest intensive therapy unit in Europe is in the QE with 100 bed place. This gives us the opportunity to have a significant pool of patients that we can enroll for our clinical studies. Another advantage of where we are working is that we are very close to the University of Birmingham, which obviously gave us the opportunity to collaborate with members from the University of Birmingham and use their expertise for our clinical studies. Finally, I would like to mention the SRMRC, which is a group that has been funded almost 10 years ago by the University Hospital Birmingham, the University of Birmingham, and the Minister of Defense, designed to help clinicians to run more studies on trauma. So not only brain trauma, also burns, hand trauma, and so on and so forth. So one of our studies was related to concussion. So in the QE, in the Queen Elizabeth Hospital, we are running a concussion clinic, and we were trying to optimize the way that we investigate the effect of concussion in acute using optics. So we try to implement FNIRS into the commonly used algorithm or neurocognitive test that we use to scan patients immediately after concussion, so within 72 hours usually, and to see if we can also see an abnormal activation of the brain. So far, this study has been limited to a pilot study because obviously the pandemic also stopped us, but we are aiming to have a more significant pool as soon as the concussion clinic Birmingham can be resumed. Obviously, we are not going to bring patients in the hospital because of COVID. So briefly, we just use 3 neurocognitive tasks. Here, the silent word generation, the digispan, and the symbol search. Here is an explanation of what was the setting, so with an examiner just next to the patients. And here is an example of the activation related to the silent word generation. We showed a higher activation in the dominant hemisphere, which is the left, as expected. Another study that we are running is on moderate and severe TBI patients. So we are using a contrast dye in the cyan and green in order to measure the cerebral perfusion on these patients, and we reported that there was a smoother curve of ICG passage on the TBI patients, possibly related to the reduction on cerebral perfusion because of brain trauma. Finally, I would like to mention one of our techniques that we developed in order to simulate on unhealthy participants a common scenario in TBI patients, which is a decrease of brain saturation and an increase of intracranial pressure. So this is the Valsalva maneuver, which is an expiration with closed glottis. Here we can have the 5 phases of the 4 phases with the normal, obviously, with the 4 phases of the Valsalva maneuver. And I ran a study in the University of Illinois where on healthy volunteers, I simulated this, I let them do this maneuver in order to simulate an TBI scenario. And you can see that there is an increase of brain saturation in the extracranial tissue, as expected with phase C, and a decrease of cerebral saturation, as expected in phase C. It also can be noted the other phases can also be seen, all the hemodynamic that follows the Valsalva maneuver also can be clearly seen in this analysis. Finally, I have to mention not only the University Hospital Birmingham, Mr. Davis, that I'm talking on his behalf, and my supervisor, Belli, but also the group from the University of Birmingham with whom we are collaborating, and also the group from the University of Chieti Pescara, in particular, Professor Chiarelli and Dr. Perpetuini. All the studies that I presented have been funded by the European Union using the funds from the BitMap project. Thank you.
Thank you, Mario. Excellent timing as well. Next, I'm delighted to introduce Jeff Dunn from the University of Calgary, Canada. He's going to talk about NIRS as a tool for studying concussion. Thanks, Jeff.
Good morning, good afternoon, good evening. Thanks to the organizers, Mari and everyone, for putting us on, and especially for inviting me. It's fantastic, I appreciate it. I'm going to introduce the lab first. So on the left, we see some of the key people that are involved in the concussion project. Dr. Hawk is actually in Boston now. And the column of my lab, some great people that are working on concussion, and also my clinical collaborators. The lab might be considered more of a fellowship, I would say, than a lab. We work well together, and here's our fellowship of NIRS. So the thread that I have in my lab is hypoxia and regulation of oxygen tension. So we do a range of projects. These are some of the clinical projects we've worked on currently and in the past. Our main focus is on MS and concussion, however, or MTBI. So the problem is big in terms of society. By age 25, as many as 25 people in the States may have, 30% of the people in the States may have had a concussion of some sort. And that every year is almost 1% of the population, so it's huge. And we decided to look at NIRS or functional NIRS because most people, or the most common symptom is they feel odd or groggy or foggy. There's some functional change that occurs. So in setting up the clinical study, we've been working on this for years now and looking at different parameters to try and optimize the differentiation between non-injured and injured people. So head cap design is huge. What regions we're going to work on. We're concentrating on coherence measurements. So interhemispheric, largely looking at these frequency bands. The y-axis here is frequency and the x is time. And there's a low frequency oscillations that link or occur in similar regions of the brain, the left and right part of the brain. If we look at fiber pairs and display them as frontal lobe or motor cortex, and red is darker or more stronger coherence, this control line along the top is controls in different functional conditions. We're varying the task and comparing to rest. And we're looking at different visualization techniques, again, to try and optimize the method. If you go over to the two-back, we're finding that during a task, we get better discrimination. So the difference between the concussion and the control is visually apparent there. This is just one person. The other things we're looking at to optimize, again, the regions I mentioned, how we group the fibers, the type of task, the length of the task, and the software that we could introduce into a clinic to make it more readily accessible. The results, some of the this is a pediatric study, our first one where we used finger tapping. We looked at motor cortex and used a TechN. On the side that we put the frequency seed, you see a small drop between the controls and the concussion. But on the other hemisphere side, there's a significant difference between controls and concussion. We done another study with the Nyrex using a two-back in the frontal lobe. Again, we see a drop in coherence between controls and concussion. The controls are orthopedic injury in this case. Some people are suggesting that the stress of having an injury itself might be some of the things we're picking up in function. So we're using orthopedic controls that have had limb injuries, as well as the to compare against the MTBIs. And this is a study in adults where we again use the two-back. This is the frontal data. We've got data from different regions, and we use the TechN. So this is all the left to right comparisons. But here is a two-back looking from the left frontal to the right motor. Sometimes not looking at the same region gives us better discriminations than looking at the same region. Now we're moving into a visualization where we're trying to identify individual people. So here the color is 2 standard deviations around the mean, and everything below that is considered abnormal. So we're able to pick out 2 populations. So in conclusion, we're discriminating against concussion and controls using FNIRS, largely with coherence measurements, and about half the people are showing abnormalities. Thank you very much.
Thank you. That's excellent. So next, we're moving on to Jana Koenig-Stolfer from Carnegie Mellon University, USA. Who's going to talk about neurovascular coupling changes with cerebral perfusion pressure.
Thanks a lot for the introduction. Yes, I want to talk about some of the work we've been doing recently on last couple of years, really, about neurovascular coupling changes and in traumatic brain injury, or rather how neurovascular coupling changes go hand in hand with increases in intracranial pressure and changes in cerebral perfusion pressure. But I thought I also say, because I never really talk about the other projects much. So the lab is really focused on like a mixture of NIRS and DCS and diffuse optical methods, like we all are doing. But we do a bit more than TBI work. So we do like EEG, NIRS, multimodality, multimodal imaging for high resolution EEG source localization with DOT. We have some really interesting progress in developing a transabdominal fetal pulse oximeter with the company. We related to this talk, mainly we've developed a non-invasive intracranial pressure sensor based on the DCS cardiac waveform changes, and using machine learning to really quantify pressure inside the head. Very related to what like Mari is doing, or Wes Baker with critical closing pressure, but also Turgut is doing with some of his DCS work. And then just because I never talk about it, but it's really a lot of fun, we also are trying to develop a NIRS brain imaging device for dolphins and marine mammals in general. But I do want to talk about intracranial pressure and neurovascular coupling and how this is related to traumatic brain injury and hydrocephalus. And so the idea is really that in hydrocephalus and traumatic brain injury, often intracranial pressure is increased. And from a clinical perspective, this is known to lead to worse neurological outcome. So optimizing pressure in the head is then in theory helping patients cognitively and with a neurological outcome. What is also known in those patients is that blood flow regulation or autoregulation is impaired. So that means that this general mechanism of maintaining blood flow inside the head at a given perfusion pressure is no longer working. So there is some sort of relationship as well. And pretty much what we're asking is if intracranial pressure has an effect on neurovascular coupling, or rather, if intracranial pressure is increased and blood flow autoregulation is impaired, what does it do to neurovascular coupling? Can we develop a neurovascular coupling based biomarker for clinical assessment? So really what we're asking is then if in the range of intact autoregulation where blood flow is maintained, we can describe neurovascular coupling as the convolution between neurons convolved with some hemodynamic response function to yield our NIRS hemodynamic signals. The question is, outside the autoregulatory range, what is that neurovascular coupling? What is the hemodynamic response function? And so we evaluated this in non-human primates where we have so the animal model is such that we can place NIRS and EEG sensors as well as DCS for blood flow, and we can actively control intracranial pressure in those animals by means of a catheter where we put fluid inside the ventricles, so we can really go up and down in intracranial pressure depending on how much fluid we put inside. So that leads to perfusion pressure changes, which is really the difference between blood pressure and intracranial pressure. And what we do with those animals is then we show visual stimuli. So it's a rotating checkerboard in a typical block design. So we have about 5 seconds of stimuli for the EEG response, and then the hemodynamic response in terms of the block design. And so what we have asked was, how does the EEG, how does the neuronal activation change and the vascular activation change with perfusion pressure? And so first, we actually wanted to know, can we reconstruct Larsen's autoregulation curve? And so that's a group average of this blood flow regulation curve in those animals. And so you nicely see that between about 60 and 100 millimeter mercury, blood flow is maintained, so autoregulation is intact. And so the very first step that we did was just plot the visual evoked response. So that's the neuronal response to visual stimuli, color-coded with perfusion pressure, and then also the vascular response. And so just qualitatively, what you start seeing is that, so blue is healthy pressure, autoregulation is intact. As soon as we go to higher perfusion pressures, visually the neuronal response is changing, and so is the vascular response. So ultimately, something is changing with pressure. And so again, what we then actually wanted to know is what is the hemodynamic response function? And so we quantified, that's preliminary work, and but we quantified the hemodynamic response function as a function of perfusion pressure. And so just qualitatively, what you start seeing is that the neurovascular coupling is actually different outside the autoregulatory range. So ultimately indicating that we might can use neurovascular coupling as an indicator of perfusion pressure and autoregulation intactness. So we're translating all of those, the DCS, NIRS, EEG systems, most of them home-built, to the clinic, and we're measuring children with hydrocephalus and traumatic brain injury, as well as starting to look into stroke patients. And so with that, I would like to thank my lab and the funding sources. Thank you.
Thank you, that's excellent as well. So next, I'd like to introduce Mamadou Diop, who's from Western University, London, Canada, and he's going to speak on hyperspectral time-resolved NIRS in the adult head.
Mamadou, we cannot hear you.
Sorry, guys.
Perfect.
Yeah, so now you can hear me.
Yes,
So thank you very much, Maria. And
but we cannot see the slides. So go back to share screen.
sorry, should be able to do this easily. I do this all the time.
Perfect. Put it in. Yeah.
Excellent. So sorry again. So I was saying that thank you, Maria, and the organizers for this opportunity. And so today I will be talking about time-resolved hyperspectral NIRS in the adult head. And these are the two students who have been doing this work in my labs. So I would start with, this is a recent paper that we published in Biomedical Haptics. And this was a study done on adult during cardiopulmonary bypass. And what you can see here in this example here is that when the blood pressure decreased, so this was a very eventful time window where the blood pressure was dropping quite significantly because this patient was having a bleed. And you can see, if you look at the blood flow, you can see this was measured using DCS, and the same with cytochrome C. And we also showed, this is something we already showed in two previous studies in animals, that the cytochrome C will only start to change when blood flow drop below 70%. So this is great, but we suspect that even we are seeing these changes. So there is a possibility that there is an underestimation of the changes. And this is a very recent neurophotonics paper that was published by Keith's group. And what they're showing here is that, so I'm going to just walk you through this very quickly. So these are the source director distance, and this is time resolved. And this is using the number of photons, which is similar to CW. And this is using the moment. First, this is using the mean time of flight, this is using the variance. And what they are doing is that they are turning, they have a pneumatic cuff around the head. And when they turn on the pneumatic cuff to cut the blood flow to the skin, you can see, especially with this, for the large source director distance using the variance, that there is a nice, so for the tourniquet on and off, there is not a significant difference. However, you can still see, this is measuring the changes in oxyhemoglobin. And the experiment was done is kind of a block design using a computerized gas system. So what you can see here is that even with this long source director distance and a large, and using the moment and with time resolve, you still have this difference. And so, and the same thing was seen also for deoxyhemoglobin. So we believe that it's because when you do this measurement, you can see that there is some variation. So you have, so your accuracy is quite large. And because you're not taking into account the spectral information, so you will be very sensitive to this inaccuracy. So we believe that by using a hyperspectral time resolve system, you can solve this problem. And we run a in silico experiment where we use MCX, and we conduct the simulation of time resolve system on an adult head using like this was an MRI image, a high resolution MRI image that of an adult head that we segmented into skin, scalp, and skin was one layer, and we have cerebrospinal fluid and then the brain. And so we simulated hyperspectral TPSF for different brain oxygenation as well as different skin oxygenation. And so we did a 2-layer fitting where we consider the extracerebral layers as the top layer and the bottom layer will be the brain. And we use a short source directed distance to estimate the extracerebral the concentration of the chromophore in the extracerebral layer. And keep in mind, in this case, we are only interested, we only simulate water, oxy, and deoxy, and we didn't yet do cytochrome C. And once we go through this analysis process, so we get these results. So what we can show here is that for the recovered oxygen saturation, we are almost bang on. The largest difference was for the total hemoglobin, and the maximum difference we measured was about 7%. And you can see for deoxy and oxy also, the results are extremely good, especially for deoxy, which is similar to what the KIF group was showing, that the recovery of the deoxy was more accurate than the one for the recovery of oxyhemoglobin. And for each of these points here, what we show here is that we have many, so these are different skin contribution. You can see the skin oxygenation, we varied from 40 to 80 and it didn't affect the accuracy. So that brings it to the end of my talk. So thank you very much. And yeah, I will be open to questions. Thanks.
Thank you, Mamadou. I'll be very interested to see your cytochrome C oxidase results when you get them. We'll have the questions at the end. Next, I'm delighted to introduce Colette McKay, all the way from the Bionics Institute in Melbourne, Australia. And she's going to talk about FNIRS to fill unmet clinical need in audiology clinics.
Hello, everyone. Greetings from the Bionics Institute in Melbourne. It's now approaching 1 a.m. in the morning, so if I'm a bit gaga, you'll know why. The Bionics Institute is an independent research institute focused on the development of novel clinical devices. So what are the unmet needs that FNIRS can help to fill? First of all, when infants fail newborn hearing screening, we need to actually assess how well they can hear so we can fit a hearing device. 10% of these infants have a condition called auditory neuropathy, which desynchronizes the activity in the auditory nerve and so prevents EEG assessment of their hearing level. So we know they have a hearing problem, but we don't know how much they can hear. So for all children, we need to be able to objectively measure speech detection and discrimination so we can validate their hearing devices. So at the Institute, we're developing automated FNIRS test and analysis algorithms for clinical use, and we're also developing a clinical hardware prototype. In the cochlear implant field, the unmet clinical needs are automated programming of cochlear implants for infants. We need to improve pre-implant prognosis of outcomes because the outcomes are very variable between people, and we need to work out how to optimize individual outcomes of cochlear implantation. We're also developing objective measure of listening effort and objective measure of tinnitus severity for clinical use. Here are our publications so far so that you can look at them later if you want. So I'm going to give you a bit of a taster of some of the outcomes of our research. The first graph I'm showing you is sleeping infants can demonstrate speech detection and discrimination. So here we see the hemodynamic responses of a single infant. This is not a group response, this is a single baby that we're looking at here. So the blue line is the response to the word she. So if you repeat blocks of she quite a few times, the response habituates. So you get this red response here to she. And after it's habituated, if you introduce a different word like see, you get this yellow response here that springs up. So the difference between the red and the yellow responses shows that the baby's brain is distinguishing between the SH and the S in she and see. So the other thing I want to show you is that babies with auditory neuropathy show a completely normal type of FNIRS response to sound. So these are the responses of from a single person. The left ear had normal hearing and the right ear had a hearing loss with auditory neuropathy. And we presented sounds at a moderate loudness level in both ears. And you can see the responses are very similar. And so we can use the FNIRS response to actually measure how much hearing a baby has if they've been diagnosed with auditory neuropathy. Going to the cochlear implant field, we've been looking at the response to cochlear implant users to lip reading, looking at brain plasticity due to deafness and cochlear implant use. So if we actually look at cross-modal activity in the auditory area of the brain, so this is activity evoked by lip reading. The more activity that you see here in experienced cochlear implant users, the poorer their speech understanding is. Interestingly, this result does not, in our hands, translate to a predictive measure. So if we measure this response, cross-modal response, early on in pre-implant or immediately post-implant, it's not related to the actual benefit 12 months later of a cochlear implant for the people. What does actually predict outcomes 12 months after implantation is the activity in the occipital cortex to lip reading. So the more actual activity that lip reading induces in the occipital cortex, the better the speech outcome. This looks like a negative correlation, but it's actually a positive correlation because this is the signal to noise ratio here of speech in noise. So good is down the bottom and bad is up the top. So thank you for listening. I'd just like to acknowledge all the funding that our lab has received from various organisations and governments, and our staff members and collaborators and students. Thank you very much.
Thanks, Colette. That's wonderful. So last but not least, we've got Anne Gallagher presenting from Université de Montréal, Canada, who's going to talk about NIRS predictive markers of neurodevelopment.
Sorry about that. Can you see my slides?
Yeah, it's showing there. Excellent.
Thanks. So first, thank you, Maria, for organizing this great event. I really enjoy learning about all this great research that our community is doing. So I'm going to talk about how we use FNIRS in my lab to study brain development, but also how we can use it to identify predictive markers of neurodevelopment. Okay, so the first study I'm going to present is a FNIRS longitudinal project that has been launched by Laura Caron-Desrochers, who is a PhD student in my lab, and is coordinated by a postdoc, Natasha Paquette, who is also lab coordinator. So in this project, we aim to characterize functional brain connectivity networks in healthy children from birth to 3 years old. Basically, we want to recruit 120 babies that will follow longitudinally. So we see them at birth and then see them at different time point for a 12-month, etc., till 3 years old. And so far we have 60 babies, 60 neonates that we recruited. And each time we see them, we do FNIRS using the NERScout device. And we also do neurodevelopmental assessment to compare with behavior. We started in 2018, and so far we have almost 200 recordings. So we are pursuing the recruitment. Our older baby just turned 24 months last week, so we just had the first 24 months babies. As I told you, a part of my research program also aims to identify early brain markers of neurodevelopment in different clinical population, in children born preterm, in children with epilepsy, but also in children with congenital heart disease. So this is a project with babies with CHD that is done by 2 PhD students, Solène Fourdin and Sarah Provost. In that project, Solène and Sarah are doing NIRS recording using the ISS machine at 4 and 42 months with babies with CHD and in healthy controls. They are also doing neuropsychological assessment at 24 and 42 months. These are very preliminary data, but these are data at 4 months from resting state functional connectivity. in NIRS. These are controls data, and here's CHD data. So basically what we can see is that babies with congenital heart disease as kind of hypoconnectivity, functional connectivity. So they have weaker and fewer long and short distance connection, and as well as intra and inter-hemispheric connections. And now we want to look at the association between the 4 months data, so these data at 4 months, and what's going on in the neurodevelopment at 24 months. So here you can see the histogram from the 24 months Bailey, scale results. So the Bailey scale gives us indices of cognitive development, language abilities, motor abilities. So basically, the CHD data are in red and healthy control data are in blue. And what we can see so far, these are still very preliminary, but as expected, babies with congenital heart disease have a language delay compared to healthy control. So this is expected because about half of the children with complex congenital heart disease will have a language delay and later on neurodevelopmental disabilities or in the cognitive or language domain or other domains. So, so that's what we see at 24 months. So now we want to look at the connection at 4 months and what are the association between the functional connectivity at 4 months and the neurodevelopment at 24 months. So in control here, we can see that there are a lot of 4 months connection that are associated with the language outcome at 24 months. However, in the babies with complex CHD, we just have a few connections that are associated with the 24 months language outcome. So we need more data to be able to interpret these results and to have stable and reliable results. But the goal is to identify at 4 months predictive marker of the neurodevelopment at 24 months or later on. So we will be able to identify who are the children at higher risk for neurodevelopmental disabilities to provide early intervention. So this is the last project I wanted to present. This is an example of how we can use FNIR-CG in clinical settings. I've been working with patients with epilepsy for many years. This is an example of a child with a refractory epilepsy. So basically we use NIRS-EG to localize the epileptogenic focus. We compare the FNIR-CG to other modalities. So this has been done with many collaborators, including Christophe Grosval presented yesterday. So we compare FNIRS-EG data to EG-fMRI, EG-MEG, and other techniques. So gathering such a multimodal combination of results can really help and guide the neurosurgeon for the epilepsy surgery in these patients who are refractory to medication. So we can localize the epileptogenic focus, but we can also localize expressive and receptive language functions, which is very important in children and patients with frontal and temporal epilepsy, which are the most common type of epilepsy. So before surgery, we want to localize language. So we can do it during a language task, of course, but also at rest by mapping a language network using functional resting state functional connectivity data. This work is done by Alejandra Husser, a PhD student in my lab, with the help of Julie Tremblay, who is a biomedical engineer, and Fetsamon Vanasingh, the NIRS-EG Tech, and the team, and in collaboration with Daniel Nguyen, who is a neurologist and scientist in Montreal. So for many years now, we have been use FNIR-CG as part of the pre-surgical assessments of patients with refractory epilepsy in our center and different centers in Montreal. And hopefully we'll be able to translate this technology that is very children-friendly to other clinical population. Thank you. Oops. Thank you very much.
Thanks, Anne. So I think we're going to move on now to question time, and Pepe Dan will lead us through that. If you can see in the messages, I've just posted the link again to post questions there on the internet there.
Okay. Okay, this is Pepe Dan from Machu University in Japan, and we have lots of interesting questions, and I don't think we can introduce them all. And let's start a question to, and actually this is a discussion, it's answered. The question to target the question is, why are you limiting the study to the periphery? Do you plan to add cerebral oxygenation and cerebral blood flow? Do you have, could you please introduce your comments to that, Dudra?
Yes. Yes, thank you for this question. So this is related to the COVID-19 project. There are two reasons. One of them is that we had to be practical in March, April 2020. So muscles were a lot more accessible, very easier to interpret. Second, there's a lot of literature actually looking at ARDS, acute respiratory distress syndrome, and sepsis by characterizing the muscle. So there's a very clear questions and very clear answers we can seek. At the moment in this project, we don't have the goal to look at the brain, but other projects are coming up soon, so things may change.
Well, thank you very much, and you seem to be very busy doing that. Then, okay, we have a question to Mario, and this is you seem to have difference of cortical activation among the trauma patients. Are there distinct difference between normal subjects and the trauma patients?
Well, our study was mainly aimed to translate FNIRS into the clinical assessment. So we didn't have time to compare our results with controls, but this is something that we definitely are going to do in the next, in the following future, since obviously we need even a bigger sample size on TBI before we can make conclusion on difference activation.
Thank you very much. And we have a question to Jeff. And actually, this is a discussion, and could you please introduce answer? When comparing left and right non-homologous regions, how can you interpret the differences between data?
Yeah, that's a good question. Thanks for that. Yes. So when people talk about interhemispheric coherence, they usually say that regions that are functioning together are coherent. So when we measure between regions that aren't really functioning together, what does that mean? That's how I interpret the question. So there's two aspects. One is we're looking for criteria that allow us to differentiate injured MTBI concussion patients from controls. So it's more of a pattern analysis that we pick that up on. And as for the functional interpretation, if we're right, and you know, the jury is a bit still out, that these frequencies link functional regions, then the frontal lobe in some periods is functionally connected to the motor cortex. And we see that a lot, actually. We do see, when you do task activation, we see changes sometimes in the frontal cortex as well. So I think there's a network thing going on there.
Okay, thank you very much. And there's a question to Yana, which is not answered. So, okay, this is about concerning the perfusion pressure. EEG signal seems more sensitive than functional neural signal. Is it really the case, and which is more sensitive?
So we're still really evaluating it. The features in the visual evoked potential are correlated with perfusion pressure, but also intracranial pressure. And it seems like the hemodynamic response function is more sensitive to actual perfusion pressure. So it seems to be the combination, but we're trying to evaluate that in a larger cohort in the clinic actually now. So I don't have quite the answer yet.
Okay, thank you very much. And the question to Mamadou, just a moment. Yes, okay. And what operation was being done in the first slide that affected MAP, BFI, SO2 so much like that?
Yeah, so thank you for that question. So these are adult patients who were undergoing cardiac surgery under cardiopulmonary bypass. So what was important here is that in contrast to FNIRS, where you know, you only need to detect an activation if it is above the ground, your noise level. In this clinical application where you want to be quantitative, it's extremely important to be quantitative. It's not just looking at those changes. So we have seen those changes, but we believe that they are actually underestimated. So that's why I believe the time resolve is extremely important and the hyperspectral approach. Yeah.
Okay, thank you very much. And then a question to Colette. And okay, yes, and concerning the visual, speech, optical log data, how do you reconcile them with earlier arguments that cross-modal adjustment have negative implications for CI outcomes? Do you see me? Can you hear me? Oh, you are
Sorry,
muted.
I was muted. Sorry,
you're muted. Could you please? Yes. Could you repeat that again?
Yeah. So in our study, we did show that cross-modal activity in experienced cochlear implant users was maladaptive to speech understanding. But many studies who do some of the studies who've done longitudinal studies who show that as a predictive factor have used prelingually deaf adults rather than our adults who are getting a cochlear implant are all postlingually deaf. And so the different degree and time of deafness actually causes differences in the way that the brain reacts to deafness.
Okay, thank you very much. And we have the question to Anne. Yes, it's about could you clarify what state the infant at each age during FC data collection, sleep, visual stimulation, and I presume you are analyzing oxy only. How's that?
Okay, thanks for the question. Actually, it depends for which project or at what age. Did the question, I don't see the question. So I'm not sure about which project you're asking, but when they are very young, the babies are asleep. For example, for the project with babies with congenital heart disease. So they are asleep, and if they have, I'm looking at oxy and deoxy signal. So we're looking at both. I'm not sure I'm answering the question.
Okay, thank you very much. Okay, so, well, thank you very much. In relation to that, well, I have another question to Anne. Can you say more about how to map the language score in the children with CHD, with the FC? Have that?
Yeah, thanks for the question. So I guess you're asking how we look at the association between the 4-month-old functional connectivity and the neurodevelopmental score or language score at 24 months. So basically, for the moment, we don't have a lot of children that reached 24 months, but so far we are looking at a correlation. So the link I showed are all the links that are significant after correction for multiplicity of testing, but these are simply correlation for the moment.
Okay, thank you very much. And we have lots of questions which is unanswered. Okay, there's a question to Jeff. And for the 2 TBA populations, do you see differences in the follow-ups?
Differences in what, sorry?
Differences in follow-ups.
Oh, follow-ups, thanks.
Yes, right.
Yeah, no, we haven't.
Sorry, I couldn't hear.
We do have a study that we're starting where we'll do follow-ups for 3 months.
Okay, thank you very much. Okay, well, there's a question to Yana. And what anesthesia did you use during the perfusion? Wow, interesting question. Perfusion pressure studies, or was there anesthesia?
So for the functional activation, we stuck to fentanyl. So we had a cohort of animals with isoflurane and then fentanyl, but isoflurane was really used as a metric to impair autoregulation. But in terms of the functional activation study part, that was all fentanyl, with really using fentanyl in order to maintain neuronal function. So it should not have an effect on the EEG signals as much, or at all, actually. So fentanyl.
Well, according to my experiment, it seems really tough to control how effective the anesthesia is, and how do you control that in your experiment?
You mean how to make sure that the animals are really
Yes, right.
So it was a mix. So we did use mainly fentanyl with very low levels of isoflurane, so really below any threshold. We periodically did toe pinch validation to see how deep the animals are. So if it's a very common way. So if there's no reaction in heart rate or blood pressure or anything, to toe pinch, there was one indicator. And so then all the other physiological parameters that we can look at, so SpO2 and EKG and such. Yeah.
Okay, thank you very much. Probably this may be the final question, and this is to Colette. Okay, I have a question regarding the habituation and novelty detection. Do you expect the novelty response to happen in slightly different regions than the first response to that stimuli? If you are really restricting the ROI to a few channels, would you expect the novelty response to happen on a neighboring but slightly different ROI? Like that?
Yes, that's a good question. I didn't explain the regions of interest. So we had some across in the auditory area and also the prefrontal area, because we were expecting the novelty response to be present more in the prefrontal area. But interestingly, in the sleeping infants, the response, the FNIRS response is very broad. It's not spatially very specific at all. And so we actually get very similar results in all the channels, even though there's slight differences. When we try the same experiment in awake adults, the responses are very localized to specific channels.
Okay, thank you very much. Okay, thank you very much. Okay, so we don't have enough time. So let's move on. Thank you very much. And we will move on to the next session. Thank you very much.
Thank you so much, Faith and David. You were awesome moderator, and thanks to the speakers. It was a really great session. I really enjoyed it. So I'm co-chair here, but I'll leave the work to Rickson, and I'll be working on the background, make sure everything runs smoothly. Rickson.
Okay, thanks, Mari. So let's start our last session. Unfortunately, it's been a great time